As Eric and Joanna were leaving the studio just now, Joanna blew my mind with the fact that bears apparently don't sleep while they're hibernating. Yeah, so Eric Palkovacs, who's the director of the Institute of Marine Sciences, guest hosts today's episode, and he sits down with Joanna Kelley, who's a professor here at UCSC, and she studies genomics, or DNA. And they talk about how animals adapt to extreme environments and how fish are adapting to climate change here in Monterey Bay. They also talk about bears and salmon and all sorts of interesting geeky science things. At the end of the episode, they have a very, very special announcement. So this is a deep dive edition of Science Solutions Santa Cruz. I'm Jonathan Hicken. Enjoy.
Welcome to another deep dive episode of Science Solutions Santa Cruz. My name is Eric Palkovacs, and I am director of the Institute of Marine Science at UC Santa Cruz. And it is my distinct pleasure to welcome Professor Joanna Kelley today. Welcome, Joanna.
Thank you. It's a pleasure to be here.
Joanna is a professor in the Department of Ecology and Evolutionary Biology and associate director of the Institute of Marine Science. And it is really my pleasure to have you here. Welcome.
Thank you.
So I want to start with, you're from Santa Cruz, which is, how does it feel to be back in Santa Cruz?
Born and raised, it feels amazing to be back here. It's a dream.
So tell me a little bit about like growing up in Santa Cruz. How was it? Did your love of the ocean, was it inspired by your childhood in Santa Cruz?
My love of nature was certainly inspired by growing up in Santa Cruz. I spent my childhood roaming the Santa Cruz Mountains. Yes, definitely had a big influence on my life. And, you know, being a kid in Santa Cruz is a dream, right? We live by the redwoods and the ocean. There really isn't a more beautiful place in the world.
Well, you're also from an academic family as well. So you've kind of have this whole career trajectory lined up pretty well. Did that influence your choice to kind of go into, you know, the field that you are in, or is that sort of a different thing?
I am sure it had an influence. My mom's a biologist and my father's a mathematician, and the work that I do is really a combination of math and biology. So I definitely had a big influence. And certainly growing up in an academic household, there's a lot of influence about encouraging going to seminars and continuing to learn and participating in academic endeavors. And so having that example definitely helped propel my career.
Awesome. Well, the deep dive episodes, if folks aren't familiar with them, are about getting really geeky about the science. And so I'm really excited to talk to you about the science today. I also will say, we have a big announcement to make, right? But I'm going to save that for the second part of the show. We're going to start out by getting into the science and what your lab is working on right now. So one of the things I know your lab's working on, you're really interested in, is adaptations to extreme environments. Can you say a little bit about what that means and what you're working on with respect to extreme environments?
So I, as you said, am very interested in how different organisms are able to live in extreme places. And my work didn't start out there. I actually started off studying human population genetics, so differences among different human populations. And that was during my PhD. And towards the end of my PhD, I realized it was very hard to get at what the potential selective pressures were that were shaping differences among different human populations, right? The human evolutionary history is very complex.
Yep.
And trying to figure out how and why different populations have different specific adaptations was very difficult.
What was the environmental differences that drove adaptation, or gene flow, a lot of movement, people moving around a lot. So there's a lot of things going on there.
Exactly. And so I realized that extreme environments are an amazing place to study evolutionary processes and adaptation. And the reason for that is that we know the selective pressure really well.
Yeah.
And so at the end of my PhD, I started working on the evolution of antifreeze proteins in polar fishes. And this was really straightforward because we know that freezing temperatures is the main thing that these fish need to deal with. And if they can't deal with freezing temperatures, they're not going to survive.
So in these polar environments, basically the temperatures are so cold that these fish would like freeze solid if they didn't have some way of coping with the low temperatures.
Exactly. Yeah. The temperatures in the polar oceans are sub-freezing, which means they're below zero. So
and
ice
yet
crystals.
Fish are there.
Fish are there, right?
And so maybe let's back up a little bit and think about how did antifreeze proteins. Like, when were these even discovered?
So antifreeze proteins were discovered many decades ago. Actually, yeah, many decades ago.
Okay.
Because people realized that fish were living in polar environments, but not freezing.
Right.
And they didn't have ice crystals in their blood. And so people started asking why. And so the early studies took the blood serum, so part of the blood, and looked at how it responded to cold temperatures. And what they found was that the freezing point was lower than fish that were not adapted to polar oceans.
And not just lower such that whatever the dissolve, like the salt, like not just a little lower, right? Such that any dissolved salts or something would lower it a little, but like significantly lower.
That's right, significantly lower.
Right.
Yeah. So let me get back to why we started working on this.
Yeah,
we thought, it would be great to work on environments where we know the selective pressure. Right. So we started working on polar fishes. And then as I was a postdoc, I learned about hydrogen sulfide and started working on fish living in hydrogen sulfide-rich environments.
Okay, so these things are kind of, you're thinking about these things together, these hydrogen sulfide springs and this polar environment, these freezing temperatures. You're working on fish sort of in both of these different places.
Exactly. And additionally, we also work on hibernation in bears. So kind of a fun addition. We have a really diverse research program in my lab, but all under the umbrella of how do organisms survive in these extreme environments, and particularly from a genetic genomic perspective.
Yeah, so that's the part that I think like your research particularly has addressed is what is the genomic basis of some of these really interesting adaptations that seem to be specific responses to these very challenging environments. So say a little bit about, the polar fishes and what you've discovered specifically about the genomic changes that have led to the evolution of these antifreeze proteins.
Yeah, so people knew that antifreeze proteins have existed for a long time, and many different fishes have evolved them, and they've actually have different genetic origins.
Okay.
So different species that have antifreeze proteins may have different antifreeze proteins, but they all function the same way to decrease the freezing point.
Okay.
And the fish that we work on is a group called the eel pouts. And there are also non-polar versions of them that live in Monterey Bay. So we're also interested in studying the fish in Monterey Bay.
So it sounds like there are multiple different sort of groups of fish that have all sort of independently evolved or come up with this solution to this common problem, which is it's too cold. You can't just be there without any unique adaptation to allow your body and blood not to freeze. So you need something, and it's evolved repeatedly in these different groups of fish.
Yes.
So I think where we're going with this is like, do they do it the same way or not?
Yeah, so we actually haven't quite addressed that. That is one of the bigger questions is how has the genome potentially facilitated these different convergent adaptations in these very different clades of fishes? But for most of my work, we've focused on the eel pouts they all have the same type of antifreeze protein. It's a shared single origin. And we've shown that definitively with our work that there's one origin. It originated a long time ago, somewhere in the North Atlantic, in close to the Arctic, but not in the Arctic, likely due to freezing conditions during cold periods. And now we've looked at many of these different species and seen that duplication is associated with differences in temperature and pressure. So
what do you mean by duplication?
Multiple copies of the gene in the genome.
So how does that work? Like, how does that duplication evolve?
So in the case of these antifreeze proteins, there seem to be repetitive elements that are around the gene. And those repetitive elements are pieces of DNA that have a often kind of short repeating structure to them. Their sequence, the letters of the DNA is highly repeating. And those seem to facilitate, if you will, the duplication of these genes.
Oh, so it's like basically making more copies because it sort of is like in the process of replicating. It's sort of like
makes a mistake,
and then it recopies it again and makes a mistake and recopies it again. And that leads to some selective advantage, presumably, when you have lots and lots of copies of this. Is that because lots and lots of copies allows you to like produce more of it?
Exactly. And so one of the hypotheses is, and the evidence shows that there are more copies in the genome in fish that are in colder and shallower locations.
Oh, interesting. Why do you think depth matters?
Yeah, depth actually seems to play a bigger role than temperature now that we've sampled more species. And we're now increasing to more individuals among species to look at this because everything had pointed just to temperature. But these fish live at lots of different depths. So that's allowed us to also dig into the role of pressure. And
Oh,
so
right on.
it seems
Okay.
that at deep pressure, there is somewhat of a protective effect, if you will, against freezing risk.
Oh, that's so interesting. So after people put away their high school biology textbook, they have to get out their high school physics textbook to follow this episode.
Very much so, yes. And also how the antifreeze proteins actually affect ice crystals is also a physics problem.
Okay, so part of the sort of the deep dive, one of the sort of themes is like, how do scientists do their work? So how do you think about like how did you just look at enough species or enough different case studies of the evolution of these antifreeze proteins to notice this pattern, and then that's what led you to this hypothesis? Is that kind of the process?
Yeah, so we knew that antifreeze proteins were present in genomes, and really early work had done some southern blots, which is a very old technique that we don't use anymore, to look at how many copies of a gene there was of the antifreeze protein genes in 3 different samples in Newfoundland.
Okay.
And at three different latitudes. And they showed from this southern blot, which is essentially a bunch of dark black lines, that there were more copies.
Okay.
And I am a trained genome scientist, so I've always thought about DNA sequencing and looking at the genome. And so knowing that there were multiple copies in the genome and that there seemed to be a gradient of copy number through the coast of Newfoundland.
Right.
I'm hopefully remembering that study correctly, that we can actually use genome sequencing techniques to count the number exactly and correlate that number with different environmental parameters.
So one of the things I'm particularly interested in is variation within species, right? And this is an example of that. It seems like differences in the number of gene copies within a species did you expect that, or like in the beginning, or was this a little surprising?
That's a great question. So that's something we're actually still looking into. So thus far, all of our data is from different species.
Oh,
And
okay.
yeah, so we've looked at a bunch of different species, polar, Arctic, Antarctic, as well as in Monterey Bay and along the coast of California and north, as well as other Atlantic species. So we, at this point, have looked essentially at one individual per species.
Gotcha.
Now-
And these are all closely related species within
well,
sort of not necessarily. Okay.
They're in the same infraorder.
Oh, okay.
So they're not that closely related.
Okay,
Yeah,
okay.
they are all related, but not super closely. Now, interestingly, if you think about our genomes, which we can think about the fish genome too, is that we have 2 copies of every chromosome. We inherit 1 copy from our biological mother and 1 copy from our biological father, right? And those 2 copies can be different in terms of copy number. So it may be that on one chromosome 1, let's say, you have a single copy of 1 gene.
Okay.
And on the other chromosome that you inherit, you can have 2 copies of that gene. Okay. So even with our single individual per species study of the fish, we can see that there's variation in copy number between the maternally inherited and paternally inherited chromosomes,
Okay. Gotcha.
telling us that there is interspecies variation. So we now have a big study of eel pouts, which are on the East Coast in collaboration with NEMAPS and VIMS, who sampled the fish for us and sent us fin clips. So we now have 300 individuals that we're sequencing their genomes to look at variation through a very large gradient, latitudinal gradient and depth gradient.
And this is, again, multiple species or
No,
one
now
species
that's
or
just
both?
one
Or
species.
one
That's
species,
one
okay.
species. That's the ocean pout.
Okay,
Yeah.
right on. So, like, if you're going to predict, so you don't have results from the study. This is like in
Not
progress.
yet. This is in progress.
So,
Yeah.
so what's your hypothesis? Let's like sort of do a little bit. What do they call? Like you write to the journal and you kind of tell them, "This is my study design. This is my hypothesis. I'm going to lay it out for you right here, and then you're going to accept my paper no matter what we find." Like, what would you write to the journal? What's your expectation?
Our hypothesis is that there's going to be a latitudinal gradient from low to no copies of the antifreeze protein all the way up to our northernmost samples, which are essentially off the coast of Maine, that will have very high copy number of the antifreeze protein.
So you do kind of expect variation within this species.
We definitely do.
With sort of latitude and depth.
And depth, yeah.
Okay. Well, because depth is a big thing in Monterey Bay. I know you are starting some new work on this question in Monterey Bay. So say a little bit about that. Like what are you guys looking at there?
Yeah, so we're really excited to study these species in nonpolar environments because, and especially in Monterey Bay, because it's literally right here.
Right, yeah. Samples are a little easier to get.
Samples are easier, and I think the questions are really interesting for a variety of reasons. But one of the things that we can ask, which hasn't been asked that many times generally because of the difficulty of identifying it, is what happens when a species no longer needs that protein.
Right.
So we don't encounter freezing temperatures in Monterey Bay. And so the species that are living here don't need antifreeze proteins.
So, this is kind of a classic question in evolutionary biology. In fact, I think even Darwin pondered things like this, like cave fish that don't need their eyes anymore. Like what happens to an organ or a feature that is sort of useless in your new environment? So It's a very classic question in evolutionary biology.
Yep. And so we are looking at that, and we see that the genes start disappearing from the genome. So they become pseudogenized. That's the word when the gene is no longer functional. So they start to accumulate mutations that are allowed, for lack of a better word, because the loss of function doesn't affect the organism.
Yeah.
Right. Whereas if you are in a polar environment and you get a new mutation that affects the function,
that's the end.
That's the end.
You freeze.
That's really bad. Right.
Right. Exactly.
I mean, this may also be why there are many copies in the genome, so that
it's
there
like an
could
insurance
be some
policy.
like an insurance policy. Although, interestingly, we really don't see many mutations that lead to loss of function in the polar environment. So we don't see anything kind of like an insurance policy. Really, they all seem to be functional and incredibly
and like
important.
Required. Yeah.
Yeah, and
right
expressed. We've looked at whether or not those genes are actually turned into RNA, and they are in polar environments. So Monterey Bay is a really exciting place for us to look at the genomes and say, how are those losses happening? What happens when it's no longer necessary? Do we get chunks of DNA removed? Do we just see a slow accumulation of mutations? And considering the process by which these genes arise, so through these repetitive regions of DNA that are around them, it's definitely possible to think that we would have large chunks go missing from the genome. I mean, that kind of sounds funny, but
Right.
or be removed from the genome because those regions are no longer necessary. And so this project, actually my graduate student, Tina Kai, was just funded to work on some of the ill pouts in Monterey Bay.
Awesome. So do you think that depth will matter in Monterey Bay? Because that's one of the things it does. We have an amazing sort of depth gradient in Monterey Bay that's sort of one of the unique features of Monterey Bay itself. Do you think depth will make a difference?
So I'm not sure that depth will make a difference for the loss of antifreeze proteins, and considering that freezing temperatures haven't been in Monterey Bay here. However, her study is bigger than just this region of the genome. So when we sequence the genome, we can look at the region we're interested in as well as the other, well, in this case, around 800 million DNA base pairs, so the letters in the genome. So they have relatively big genomes, and we can look at the rest. And so we're really interested in looking at differences between populations at different depths. And we expect to see potentially changes that are correlated with depth, especially in genes related to vision and
Okay.
sensing, right? When you think about depth, you have to
Light.
remember exactly it's also darker
Yep.
in the depths. And so genes involved in vision, any light sensing, we expect those might be different. And so Tina can actually look at this.
If you haven't visited the Seymour Marine Discovery Center at UC Santa Cruz in a while, or ever, you really should come visit us. We are right on the bluffs of the beautiful Monterey Bay, with an aquarium full of local species and a bunch of hands-on science exhibits that help you understand what's happening in our ocean and along our coast. We are connecting the incredible research happening at UC Santa Cruz with real-world solutions for our coastal community. We don't want science to just sit on a shelf. We want it to shape decisions, inspire kids, and help our community feel equipped to act. So if you care about the future of our ocean and our coastal community, or you just want to experience a place that makes you feel a little more connected to it, come visit us at the Seymour Marine Discovery Center. Science Solutions, Santa Cruz.
So you have this like amazing window into all these different potential adaptations to all these different environmental drivers, which is really, really fascinating. I mean, one of the things that sort of the relevancy of kind of this work and why it matters, I think about things like, well, polar environments are warming, right? And so as polar environments warm, is this a new sort of selective environment now? Is what's going on in Monterey Bay with sort of the potential loss of antifreeze proteins sort of also starting to happen in Newfoundland where you studied it before? Is there the potential for contemporary selection and evolution in these populations as polar environments warm?
Yeah, I mean, I think the answer to all of your questions is yes. But to elaborate on a little bit, one of the interesting things in comparing our polar species to Monterey Bay is we can ask how the genome responds to loss of a selective pressure and new pressures, because actually the Monterey Bay species are the newer species.
Okay.
And so we can ask what happens when temperatures get warmer. What happens to these genomes when temperatures change, specifically when they actually get warmer? And so that gives us a really good picture about what could be happening to species at both poles. Now, the pace of change is so different, it's hard to determine whether or not we would see those similar changes because the pace of change and effectively the selective pressure is so much greater now than it would have been in the past. But it does give us a window into making predictions about how the genome would change. Similarly, looking at cold adaptation, or on the flip side, which we don't look at in my lab, but we think about is warm adaptation and species adapted to warm temperatures.
Right.
Because that can also help inform what species may do better in certain places in a warming climate.
Yeah. So, I mean, that's, I think, a big question is, are we going to see large scale sort of adaptation of what currently exists in a certain place? Or are we going to see replacement of those species with others that are already adapted to the conditions that are going to be sort of new in that place, or some combination of those things? And I guess another piece of this is, to what extent is this going to be adaptation of the populations in that place versus sort of gene flow or the movement of organisms from other places that may be sort of, hey, look, I've already lost my copies. Now there's no barrier for me to like invade this new habitat. Like, we're moving north now. We have this new opportunity.
Yeah, I mean, I think we're already seeing that second case, right? We're seeing lots of different species moving northwards. And as populations that are not adapted to warmer conditions or more variable conditions, whatever it is, they are reaching their limits, right? And we can think about that on the coastline. We can also think of that is also happening in alpine situations, right? As going up mountains.
Right. And the thing about mountains is sort of like you get to the top and you can't go any higher,
That's right?
And so in some ways the the North Pole is kind of like that too, right? I mean, kind of at a certain point when we have ice-free summers,
That's right.
it may be very invadable by pretty much anything. You don't have to have these adaptations anymore to make it eventually, right? If the world keeps warming at the pace it currently is. What do you think about the conservation status of these fish that you've been studying that have these unique antifreeze proteins? Do you think we should be worried about their conservation given global climate change?
I think that we should be worried about the conservation of all organisms, so including the fishes. But given the scale and pace of change, I think that we should be thinking globally about the impact of change on all organisms.
It seems like the history of the sort of really interesting evolutionary history of these organisms is kind of part of what's important to conserve, right? Is the fact that they have evolved these cool, unique adaptations. And it's like, wow, we don't want to lose that evolutionary legacy.
Certainly. And, the fact that when these species are outside of polar environments, they lose those genes
Right.
suggests, in addition to them not being necessary, they may actually be maladaptive, right? They might have a negative consequence in a warmer, non-freezing environment.
Right. Okay, so we've talked a lot now about polar fishes, but I do want to talk about bears. You mentioned bears, also interesting case. We haven't really ever talked about your bear work before, so this is going to be brand new conversation.
Right. Yeah.
So hibernation in bears is what sort of I think you've worked on a little bit.
Yep.
And I guess my own personal interest in this, I'm just going to ask questions from my own perspective of interest, which is the Fat Bear contest.
I love Fat Bear Week. Yes,
Fat Bear
Katmai
Week.
National Park.
Exactly.
The bears
So,
are amazing.
of course, I'm interested in these bears that eat salmon, right?
Yes.
And they like put on a massive amounts of weight by eating salmon. And thinking about, what is their hibernation strategy if they can put on this huge amount of weight that sort of like maybe interior bears don't have the opportunity to do? Do you think that has impacted their hibernation strategies or any adaptations that they have or even behaviors that they have in terms of hibernation?
Yeah, that's a great question. I don't know. I mean, the interior bears also put on a lot of weight.
Okay.
Right? So maybe
Did they ever win Fat Bear Week though?
Well, they're not part of the competition because Fat Bear Week is only at Katmai National Park. And I don't know that they would win because the bears at Katmai are
They get
huge.
really chubby.
They get really big. Whether or not that has led to differences in the adaptation of hibernation in terms of how much fat they put on, I'm not sure. But all bears need a lot of fat to survive through the winter. Now, those bears have this very easy access to fish. But historically, there have also been salmon runs that go pretty far interior.
Yeah, absolutely.
And of course,
once upon a time,
damming
right?
and other things have changed that a bit, but bears are very resourceful. They're very food motivated and will find food, right? And this is of course a concern for people and bear interactions because lots of the issues with human bear interactions is because people leave their food out, right? This is the biggest issue in any place. Like you think about black bears in Tahoe.
Right. Oh, totally. They're like in everybody's trash.
That's right, because people don't secure their trash. And so we are very responsible for that. Bears are very resourceful. And if you leave something out or leave something cracked, they will get it.
Yeah, like the pizza you put in your trash is like
That's
a perfect...
right. You leave it in your car and the bear will
and
just
like, yeah,
open the door
exactly.
and get the pizza.
The door of your car is like the candy bar wrapper.
It's like,
They'll
no
peel
problem.
it right open.
Yeah.
Totally. So I think another thing about these coastal bears, though, is that their hibernation period is a little shorter when they have access to these large food supplies. So that, and also, I guess maybe going back to climate change effects, right? Maybe winters are also not as severe as they once were, getting shorter. Do you see changes in hibernation season for bears?
So my group hasn't looked at that directly, but there's definitely a gradient of how long bears hibernate latitudinally as well, right? So you think about how long, cold, and dark a winter is. It is not as long, cold, and dark in northern Washington as it is in Alaska. And so bears in Washington, or bears that were in Washington, will hibernate for shorter duration than bears that are much farther north.
Okay.
Right. And it's all of the temperature, day length signals that indicate to go into and out of hibernation. Now, of course, warming is a big issue
Right.
because bears are getting the signal to exit hibernation earlier. Wake up. Well, they're not asleep during hibernation. We can talk about that in a
Okay,
second.
yeah.
But,
You should clarify what are they doing? What is their mental state?
But so finishing the first thing is that, when bears come out of hibernation, they're obviously going to start foraging, which is looking for food. And if there's a mismatch between when shoots and new leaves are available for them to start munching on, because that's what they first eat, and when they come out of hibernation, there's a big issue.
Yep.
Right?
Yep.
And if bears are near to humans, then they're going to start looking in places that smell delicious.
Yeah.
And so I think the additional concern about climate change and hibernation is this mismatch between when animals are coming out of hibernation and when the food that they rely on to start eating and continue eating through the season are not matching anymore.
Right. I mean, there's these weird periods that are sort of, so I have family members in Minnesota, right? And in northern Minnesota, winters used to be, it got cold, it would freeze. It stayed very solidly frozen, sort of historically, through the entire winter. There was not a thaw until actually spring arrived. More recently, there are these sort of weird midwinter thaws. Is that sort of what you're talking about? Is if there's this early thaw and then it even could get cold again, but now the bear's sort of not awake, but whatever, aroused to whatever you call the non-hibernating state. It's like, now, okay, but then it gets cold again and it's like, whoa, winter's not really over yet.
Exactly.
Yeah, well, the bears are super cool. And of course, California, there used to be, California grizzly bears. Can you speculate a little bit about what the ecology of California grizzly bears was like? And do you think they hibernated? California grizzly bears hibernated?
So that's a great question. And I've thought a little bit about it. I mean, something amazing is that there were brown bears in Santa Cruz. I love the stories about brown bears in Santa Cruz. There's a Mountain Charlie Road Mountain Charlie had an encounter with a grizzly bear and then had briefly a metal silver plate on his head because of the large gash that the bear put in his head. And that was then removed because he was having a reaction to it, and just for the rest of his life had a big gash in his head because of this encounter with the bear. But and Mountain Charlie Road is in Santa Cruz.
Right on. So, I mean, Santa Cruz Mountains had bears. And like my understanding is also that the bears in this area were very coastally oriented too.
Yeah, so I didn't actually answer your question about hibernation. So, food, if you think about Santa Cruz and food availability, there isn't a huge change in food availability in Santa Cruz. And so if I had to guess, I would speculate that brown bears in Santa Cruz, if they did hibernate, it was a very short hibernation period, if at all.
Okay.
Yeah, because it's really about food availability.
Right.
And bears that have access to food will not hibernate. Right. And this is, again, why it's so important to put food away. I feel like a public service announcement of if bears have access to food, they won't hibernate.
Right.
So
okay.
if you feed bears through the winter, they won't hibernate. So I wanted to tell you about hibernation.
Yes.
Right. So the bears are not asleep.
Okay.
Right. And I learned this, admittedly, in my 30s. Because I thought they sleep all winter, right? It's like there's so many kids' books, baby bear won't go to sleep, right? We're taught that hibernation is sleep.
Right.
But it's metabolic depression.
Okay.
And they actually do still have a circadian rhythm.
Uh-huh.
It's muted, and they have sleep-wake cycles.
Okay.
Which is kind of cool.
So they're kind of awake and hibernating, and then asleep and hibernating, and then awake and hibernating.
Yeah, it's just there. It's just muted. But they're very metabolically depressed. And so they are not moving, right? They don't urinate. They don't defecate. They're not eating. They're not drinking. And they're really not moving. Right? They'll rustle around a bit, but there's really very little movement.
And so when what are the cues that generally they're using when they sort of, aroused from hibernation? Like, is it temperature? Is it something else that they're detecting in the environment? Like, what causes them to come out of the hibernation state?
I'm not totally sure. But I think that it's a combination of temperature and light changes.
Does it have anything to do with like the amount of sort of reserves, like energy reserves they have?
Honestly, I'm not sure about coming out of hibernation, but certainly going into hibernation, the amount of reserves has an effect on when bears enter hibernation. And if they don't have sufficient reserves, they'll actually try to continue foraging to put on more reserves. And if bears don't have sufficient reserves, they will die during hibernation.
Yeah.
Right? Because what happens is they go from, eating food for fuel to burning the fat for survival, right? And so if you don't have enough fat on your body, you won't survive as a bear.
Okay.
And so that's a really big deal. Also, if females don't have enough fat, they won't give birth. So bears actually give birth during hibernation. They have delayed implantation.
That's fascinating.
Yeah. So they'll mate during the active season, and then there will be delayed implantation. And so if a female does not have enough fat reserves, they won't implant and they won't give birth. And actually,
interesting.
larger females have larger, healthier cubs.
And those cubs, basically, the mama bear is hibernating. The cubs are born. The cubs are just got to figure it out.
Yep, they're just moving around, nursing. Yep, and mom is just laying there. But that also means that she has to have enough reserves to also produce milk.
Yeah, right on.
And be able to provide sufficient nutrients for those cubs. And there are often 2 cubs that are born. They can have up to 3.
All right. Well, you have got a ton of awesome stuff happening in your
There's
lab,
a lot of stuff
obviously. But there's even more awesome stuff coming down the line. So this is a big announcement. So what is the big, interesting new thing happening?
Yeah, great question. I am very, very excited to announce the new Center for Marine Genomics at UC Santa Cruz.
That is awesome. Congratulations. So I am very, very proud to say that I'll be part of the Institute of Marine Science. So we're so excited. So tell me a little bit about why did you want to launch this new center? What was your motivation for doing this? You got awesome lab stuff going. You wanted to take more stuff on?
Absolutely. Yeah. One of the things that drew me to Santa Cruz, in addition to being home, was the just, international reputation for the Genomics Institute and the Institute of Marine Science.
Yeah, these are really 2 of the crown jewels of UC Santa Cruz.
Yeah. And so being someone who works on genomics and marine organisms, for me, it was just such an exciting opportunity to come to Santa Cruz. And so when I moved here, I was really drawn to figuring out a way to bring those 2 crown jewels closer together even, right? And the work in the Genomics Institute is so incredible. And the work that's coming out of the Institute for Marine Science, incredible. And so I thought, hey, what we don't have is a center for marine genomics, which being in Santa Cruz seems like an incredible opportunity.
Absolutely.
And I am at a point in my career where I was excited to take on this opportunity to bring research together and training opportunities to our students and our researchers.
Yeah. So what are some big goals you have for the center?
So one of the big goals that I have for the center is to bring people together who are working in this area. A lot of us are doing this or serving on committees and providing some of that genomics insight into that. And so really building the community and showing everyone, hey, here are all the people working on it. And ultimately, as I know is a goal of the Institute for Marine Science as well, of bringing the larger Monterey Bay community together around this, right? We have such incredible science happening along this portion, along the California coast wholly, but definitely, in the Monterey Bay, just bringing all of us together and creating that community.
Yeah, that's awesome. So genomic technologies now can be sort of widely applied to answer a whole variety of different questions in marine science. Are there certain things you're interested in tackling in particular, or in terms of scientific questions, or in terms of training opportunities?
Two of the things that have me really jazzed is providing opportunities. So I don't want to drive the science forward. I want to have the center be able to provide seed grants, which of course we need additional funding to be able to do that. But really a vision is to be able to provide seed grants to bring people together and use these genomic technologies who aren't necessarily using them. The other thing is that marine organisms are very diverse and have all sorts of challenges when thinking about DNA extractions or making libraries. It's very hard to get DNA out of some of these very gelatinous organisms.
Jellyfish, it's hard to get DNA out of jellyfish?
I haven't tried with jellyfish. I know it's sometimes hard to get DNA from kelp, for example.
Okay.
And so having this community where individuals who've already tackled a challenge, turns out the eel pouts, it's very hard to get high molecular weight, long DNA from these fish. And so we've done a lot of work just putting the methods together to optimize how we do things. And so, being able to share that knowledge.
Share knowledge, techniques.
So we want to be able to provide seed grants for people who really want to combine their marine science with genomics, as well as provide training opportunities for students. So one of the other big goals is to provide workshops for students who are maybe in a lab that doesn't have a big genomics component. Right. I serve on a lot of your students' committees.
Yeah, you're on, I think, the committees of almost all my students, because my lab sort of dabbles in this stuff. And so we have a lot of questions, but we're not experts in the tools and techniques. So it's been amazing for them to have you on their committees. And this would be great to expand the opportunities for this kind of thing.
Exactly, for the opportunities for students, because, I can't be on every committee as much as I do say yes a lot.
You're on a lot.
But having really some kind of formal training opportunities for students. And I'd love to be able to offer kind of a marine genomics summer course. That would be incredible. I think, we'll start off with a launch in probably October, where we bring the community together. And also really serving the community and figuring out what do people need. I know that students want additional training opportunities. And of course, research funding is always an issue and becoming a larger issue. So the ability to hopefully provide seed grants. So we'll do definitely fundraising to, be able to expand the mission of the new center, but it is really exciting.
Yeah. And I mean, one of the goals of Institute of Marine Science more broadly is really to bring people together to facilitate large scale proposals that are interdisciplinary. And the center is so exciting because it provides this whole new realm that we can sort of take advantage of. I'm so excited about it, and so excited for you that this is this will be great. It'll be awesome.
Yeah. And as you said, bridging also to the Genomics Institute, there's just the amazing, telomere to telomere genome work that's happening there. So much biomedical work, conservation work, and being able to bridge the knowledge that's gained there to the marine sciences.
Absolutely. Well, congratulations. Such an amazing opportunity, and you're so well poised to make an impact. It's such a good thing for UC Santa Cruz broadly. It'll be really exciting. Thank you so much for joining us, Joanna Kelley. This is Eric Palkovacs, and we are wrapping up Science Solutions Santa Cruz. Thank you so much for watching and listening.
All right, that does it for the pod. Thank you to Eric for guest hosting and to Joanna for joining him in the studio today. Congratulations to you both on launching the Center for Marine Genomics and for announcing it right here on the show. Thanks to everybody at the Science Division for helping get this incredible project off the ground. And of course, thank you to Edith Belmont for editing, Carrie Goodbar for production, Valerie Situ for distribution and socials, and thank you for listening. We'll see you next time.